northern southern blot, and there are others, I kid you not, but no matter what direction is in the name, the basic premise is the same. Take some biomolecules and separate them, typically by size, then transfer them to a membrane and probe to analyze. If the probe finds a match, a band you will see, but what kind of molecule will it be? If it's a western blot, then you'll be looking for protein. If it's a northern blot, you'll be looking for RNA. And if it's a southern blot, you'll be looking for DNA. And you use different types of probes to find these different molecules. With the western blot, you're going to use antibodies recognizing the protein. With a southern or a northern blot, you're going to be using short little labeled nucleic acids, typically little labeled DNAs, often with a radio labeled phosphorus on the end or some sort of fluorescent tag, and that has a sequence that matches the sequence that you're looking for. And so these are just 3 of the most common types of blotting. There's also a bunch of different variants. We have Eastern, reverse northern, all sorts of different ones. So I'm going to go most into detail about the 3 major ones. So we have our western blot, where we're using antibodies to look for proteins. Our southern blot, which is the original blot, and the southern actually comes from the name of Edwin Southern, who actually invented this technique in the 1970s, and then the other names are just kind of like puns off of that one. So only the southern actually has the capital S. The rest have like a lowercase letter, just technical wise. Okay, so we have western blot, lowercase w, and antibodies looking for protein. We have southern blot, we have DNA looking for DNA. We have northern blot, we have DNA looking for RNA. And so let's go more into detail, a brief overview, compass of the blotting directions, and we'll also talk a little bit about mentioning the other variants, because the basic premise behind all of these is the same, that you're separating things and then probing for something, and it just can vary based on what you're probing for. And importantly, we're looking for really specific things. So we're looking for like protein A, but not protein B, and we're looking for DNA sequence X instead of DNA sequence Z, and we're looking for RNA sequence Q instead of RNA sequence R. And so basically we're looking for very specific things, so we're kind of going specifically fishing with bait that only matches the type of fish that we want. Okay, so I'm going to start with an overview, some similarities and differences between these different methods, as well as some of the reasons why you might want to use various ones in different situations. And so, for example, although we use western blots a lot still, for northern and southern blots, a lot of the uses have been like superseded by PCR-based methods. So with PCR, we can basically make copies of specific sequences using sequences that are specific for those. But there are still reasons why, in some cases, we will still use northern and southern blotting, because we can get different types of information that we couldn't get with PCR. And so we'll get more into those in them. And I just want you to know that these still are techniques that are used, even if they're not used as frequently as they were in the past. And we'll also talk about some of their past uses, which are really important in pioneering genetic studies. So we're going to be looking for protein in western blot, and RNA in northern blot, and DNA in southern blot. So it turns out that it's a lot easier to look for DNA or RNA because of their sequence specificity. So the reason why DNA is double-stranded is because the DNA bases, so those unique parts of their letters, have this complementarity, the specific base pairing. A to T and G to C, and then in RNA you have A to U. So RNA has uracil, thymine is in DNA, but they both can bind to A. And so you can get RNA to bind to RNA, RNA to bind to DNA, DNA to bind to DNA. And so in these experiments, what we typically do is we use a probe that has a sequence that is complementary to the sequence that we want. So if there was a G in our sequence, we'll have a C in our probe. And note that it can get kind of confusing, because the strands are what we call anti-parallel. So you have this running from 5' to 3', and this one running from 3' to 5' if you're looking at it this way, but we typically write the sequences from 5' to 3'. So our probe is going to be like the reverse complement of the strand.
In the probe, we're typically using a DNA probe because DNA is more stable, which is also going to come into play with some of the differences between northern and southern blotting. So we can make a probe that has a sequence that is reverse complementary to this sequence that we want to look for. So it's going to stick to the thing that we want. We, of course, need to make it long enough so it's actually going to specifically stick to the thing that we want, kind of like a password. The longer it is, the more likely it is that no one's going to guess it just by chance. And so if we use a longer sequence, then we have a better chance of just finding the sequence that we want. But then we need to wait a way to see it. And so we're going to see it by, typically we radio label the probe. And so we stick a radioactive phosphorus group in the end, a phosphate group. More on this later, but this is going to be super duper sensitive. You can also, in some cases, use a fluorescent tag to avoid the radioactivity. So we have a way that we can detect nucleic acids, but how about proteins? So proteins are a little trickier. We don't have a direct like sequence to sequence thing with proteins. Instead, we have to rely more on the protein's shape and the various amino acids. So the various protein letters have these different features. And so they have unique like binding sites that can be recognized by little proteins called antibodies. And so antibodies are just these little proteins, and they have this generic part, this kind of adapter part that is, well, it's only generic for the animal that made the primary antibody. And then it has this variable region that is unique. So this is the constant region. Remember, it's constant for that animal. And then you have this variable region, and this variable region is going to be specific for the protein that you want. And so you can use this antibody in order to probe for the thing that you want. But you also, now we need a way to detect it. Typically how this is done is that we have a secondary antibody. And so this secondary antibody is going to be attached, or what we say conjugated, when it's like permanently attached like this. It's conjugated to something detectable. So this can be either a fluorescent compound. It can be something that can be converted to a colored compound, such as with like an HRP system. And I'll get more into this stuff later. But the secondary antibody is going to bind to the primary antibody. So it's going to bind to the generic part of the first antibody. So say this was like, our protein is X. And so this would be an anti-X antibody, but it would be made in some animal. So it could be like mouse anti-X. And then this antibody would be against the mouse antibody. So it would be like goat anti-mouse or something. By using the secondary antibody, you're making this cheaper because you don't have to have this one conjugated. You can have this one conjugated, but not this one. And so this can be cheaper. And then you also have this signal amplification, because you are adding you can now have multiple antibodies binding to that primary antibody. So there's more binding sites, and that's a greater signal. Even though that secondary antibody saves you some money because you don't have to have this one like specially made to have the conjugated part on it, this step can still be a problem if you don't have an antibody that is specific for your protein. So actually making these antibodies is not nearly as simple as just ordering a short little piece of DNA or from a company, which can be super duper cheap. If you've ever done PCR and have ordered primers, you'll know that that's probably one of the cheapest things that you can actually buy in a laboratory. But making an antibody is a whole big process, and it can be very expensive. Typically involves animals. Although there are other methods using yeast and stuff nowadays, very cool stuff on the horizons, and in starting to be used more. But it's going to be a long, expensive process. So often what people do is if they have a protein that they're expressing recombinantly, so basically they put the instructions for making the protein in there and then they make it, or they add the tag somehow into the version that's already in the cells or whatever you're looking with some like gene editing technique. You add some sort of a tag, like maybe this is just like a tag, I just mean like a short sequence of amino acids at the end, maybe like an HA tag. a flag tag, and then you can use an antibody against that tag. So say you could have this star was my tag, this could be like a flag tag protein or an HA tag protein. And then the antibody, my primary antibody, would be an anti-flag or an anti-HA or something. And those antibodies are going to be a lot cheaper to get than if you want to have an antibody custom made for the protein that you want. So now we have our ways to probe. So if we want to look for protein, we're going to use antibodies. And if we want to look for a nucleic acid, we're going to use typically DNA because DNA is more stable than RNA, and DNA can bind to DNA and DNA can bind to RNA. And we can make these specific sequences to look for specific pieces of DNA and of RNA. And so now let's go into how we're going to actually do these techniques. So the first step in any of these is going to be to, well, not quite the first step. The first step is going to be to isolate the thing that you want. So typically we're starting often with some sort of cells, and we're trying to see what's going on in cells. And that's why we have to go through all of these things to actually like fish for various things, because there's a lot of stuff going on in cells. It's not like we just mix a couple of proteins that we purified. We are dealing with this whole mixture, and there's so much stuff in there. If we were to just like do a generic stain or something to see all the proteins that were there, or to see all the DNA that was there, or all the RNA that was there, it would just be like big giant smear. And instead we need to, and we'd have to load so little of it that the amount of our thing of interest would be really small, or else we just like block the gel wells and they wouldn't separate at all. And so we're having these tiny amounts that are in a sea of a lot of different molecules, and we're looking for very specific molecules. Even though we're having these really specific or this really sensitive probes and stuff, we're still dealing with, if we had everything just in one clump, it'd be really hard to see like, was our thing actually there? And also it's good to have some sort of separation step, not only to make it easier for our probe to find things, but also to have a second like validation that the thing that it's actually, that the signal is in the spot that you would expect it to be. So if you know what size of something you're expecting a protein to be, and then if it runs in that size on your gel, then it would be like a little bit of a confirmation. So let me just, getting ahead of myself. Let me talk about these gels. So the basic idea is we're separating we have this big collection of molecules, and we want to separate them by size. And we can separate things by size using gel electrophoresis. And so gel electrophoresis is a technique where we make a gel mesh. So on the various things, and then we separate molecules by size, because these molecules are traveling through the gel mesh, and the bigger ones are going to get tangled up more, and so they're going to travel more slowly. And so when you turn off the power, so we're getting them to move through the gel using electricity, so why we call it electrophoresis. We have an electrical gradient that they're motivated to move through. And then when we turn off the power, they stop getting motivated to move. And so they're going to stay where they are, and they're going to be separated based on their size. The types of gels that we can use are going to vary based on the molecules that we want to separate. With proteins, we're typically doing an SDS page gel, although you can also do one of these, a Western blot with like a native page gel. With SDS page, you denature the proteins you unfold them and you coat them with this negative charge detergent. So it's like soap called SDS, and this is going to give them the negative charge that they need to be motivated to move through the gel. And it's also going to keep them unfolded and allow them to kind of slither through the gel instead of just traveling based on their shape and stuff too. So this is going to separate them by size, and we do it in these vertical slabs, these thin slabs. If we want to deal with DNA or RNA, here it's going to depend on the size of the molecules that we want to separate. Sometimes if we're dealing with big molecules, we can use an agarose gel. So this is one of the typical agarose gels you probably do in like labs that you might do in school or that sort of thing. These are the ones that we use in our lab most of the time, these small ones. But for a lot of cases, when you're doing this sort of high intensity southern blotting stuff for northern blotting, whatever, you're using a bigger box. So this is an example of a bigger gel box. So same type of thing, but on a lot bigger scale. If you have smaller molecules, you can use a urea page gel. And so this is what I use most of the with my RNA, because I'm dealing with small RNA. So the ones I work with are about 20 nucleotides long. And so the urea page, it has a tighter mesh. And so it's going to separate the smaller things better. And again, it's in a format kind of like an SDS page, except we often do it in a bigger size, and we can do it in an even bigger size than these ones as well. In order to get the molecules ready to move through the gel, we need to first do a couple things depending on what the molecules are. And so in proteins, we do the SDS coding. And that's going to unfold them, and it's also going to give them the negative charge that they need. When we're dealing with DNA and RNA, they're already negatively charged, but they also have structure we need to get rid of. So when we're doing a northern or a southern blot, remember we're using probes that are going to recognize specific sequences. So we need to make sure the specific sequences are visible. They're not going to be visible if they're hidden by another strand. So if you have double-stranded DNA, you're going to have another strand blocking the binding spot. RNA is not typically double-stranded, but it does fold up on itself and make all these hairpins and these cool structures. These structures are, one, going to interfere with the movement through the gel, and two, hide the binding sites. And so we don't have to worry about charge. So one of the reasons why we add SDS to proteins is to give them a negative charge. We don't have to worry about with the DNA and the RNA because they already have a negative charge, but we do have to worry about their shape and making sure that the site is going to be visible. So typically with a northern blot, so when you're dealing with RNA, you use like formamide, which is going to denature the RNA. Remove its shape, make sure that the sites are not going to be hidden. With a southern blot, you typically use denaturing. You do the denaturing with basic or alkaline conditions, which is going to separate the DNA strands. So first off, you would not want to do this with RNA because there are alkaline conditions that could actually hydrolyze. So the 2' OH that makes it RNA and not DNA, it actually also makes it liable to attack itself and break. And this is going to be can happen more under basic conditions because under basic conditions, this is more likely to deprotonate and then attack itself. And so you wouldn't want to use this with DNA, or with RNA, but because DNA doesn't have the OH, you can do this. And so you can use these alkaline conditions. So you can use these alkaline conditions to kind of deprotonate bases and various things in order to get the DNA strands to come apart and show those sites. You can do this several different places depending on the technique. Some of them do it in the gel, so you use like an alkaline gel. Some of them you soak the gel after you've run the gel, you soak it in a basic solution. Others, you soak the membrane in a basic solution after you've done this, often by the basic solutions like NaOH. And then these steps have a neutralization step where you add like acid or something, I mean, not like strong acid or anything, but to neutralize the solution before you move forward. But in these cases, you're denaturing so that you can have the probes bind and not have to worry about that stuff, as well as have them travel the size that you want, that you would expect them to be when you're talking about a northern blot. Speaking of denaturing, one thing about the Western blot is that the antibodies, you need to make sure that the antibodies you're using are going to recognize the denatured proteins. Sometimes antibodies, if they're made for detecting the protein, like in cells or something, they might recognize the folded form of the protein. And with the Western blot, you need to make sure that your antibodies are recognizing the unfolded form. And so you should be able to see that information when you look at the website or whatever for the antibodies. But a lot of times they have antibodies that are made for Western blotting. So with our southern blot, we have a couple of things that we need to worry about with the DNA. So typically when we have DNA, we're taking like genomic DNA. So we have these big old pieces of like chromosomes, which is all this coiled up DNA, tons and tons of really long thing. We need to cut it up before we can actually do our probing. And so what we're going to do is we can cut it up using restriction enzymes, so these sequence-specific DNA cutters. And so we use ones that have a fairly generic-ish sequence, so it's going to cut it at. So there are ones that are typically used to give you a size that is going to be compatible with the technique. So you cut it up into those pieces, and then you are going to separate them using your gel. And so now we've run our gel, and so whatever type of gel that we've run, we've stopped it, and now the molecules are stuck where they are in the gel. But we need to be able to probe them, and we need to keep them from running away. So although they are kind of trapped in the gel because it's this mesh and it's hard to move, they're going to be able to wander a little. So diffuse, just kind of like randomly move around. And if they even though if they can't move far, they can move a little bit. Plus these gels can rip. Plus we need to have this way that we're able to probe them. So end result, we need to transfer them to a probable membrane. And so the type of membrane that you're going to use may depend on your application. With a Western blot, this could be nitrocellulose or PVDF. With a Northern or Southern, you're typically dealing with nylon or nitrocellulose. These membranes are just going to be kind of things that we can put the molecules on and get them to stick, and then be able to work with them and probe them. And so how you do the blot, there's different methods. With Western blot, what I typically do is just like this semi-wet or this like wet transfer blot. And so basically this is an electroblotting technique where you use electricity. And so before we were using electricity to send things going down through the gel, with this electroblot, we're taking the proteins out of the gel and onto this membrane. And so now we're going in the other direction out of the gel and onto the membrane. And we can do this by making this like sandwich, and I have more on Western blots and other posts, but this way you're basically transferring the proteins onto the membrane. To transfer DNA or RNA, what you often do is you just do it by using capillary action, so wicking. So you take your gel and you put your membrane on top, and then you put a bunch of paper towels and some sort of weight. And so what's going to happen is that through capillary action, so the same thing why when your paper towel gets wet, the water like wicks up, the water is going to want to wick up. And it's going to want to wick up through these paper towels. And so, but the liquid is going to get wicked up, but your RNA or your DNA in a Southern blot, it's going to get trapped on the membrane. And so it's not going to get to move all the way. And so it's going to get stuck on this membrane. And there are alternative methods where you can use some form of electroblotting, or you can use some sort of vacuum to try to get the molecules to move more quickly. But this is the conventional method that's done, and you can do this with your DNA, or you can do this with your RNA. If you are doing, this works with the agarose, but if you're doing like a polyacrylamide gel, then you typically need to use some sort of electroblotting because the pores of the gel are finer. So at this point with your Western blot, your proteins, they stick pretty well to this nitrocellulose membrane. Your proteins are all denatured and stuff. You've removed the SDS. Now these proteins are all unfolded and they have all their hydrophobic parts. So the parts that don't like water and stuff, they're all exposed. And so they're going to glob onto this kind of this membrane that you have them on, and they're going to stick okay. But in the case of DNA and RNA, you typically want to somehow lock them in place to actually get them to stay still better. So you can do this with a couple of different methods depending on the type of membrane. So if you have a nylon membrane, so nylon is positively charged. With these membranes are typically, you use like a positively charged nylon membrane. And so that's going to be attracted to the negatively charged DNA or RNA, and so it's going to stick, but not through like covalent bonds, just through temporary bonds. But then if you add UV light, so you put them in one of these like UV ovens, then this can actually induce covalent bond formation. So basically you get the permanent bonds formed between your nucleic acids and the membrane. And so you can use UV for nylon membrane. If you're using, if you're using like a nitrocellulose membrane, you can heat it up. That's going to kind of get them to shed some water, expose their hydrophobic regions, and clump up more and like cling more to the membrane. And so you can like bake it, and this way you're immobilizing the thing that the nucleic acids you want to probe for. Now we need to somehow block the things that we don't want. We need to block the background. So when we're doing a Western blot, we need to worry about the antibodies just kind of binding to the membrane, because remember, these antibodies are proteins too. We have this membrane that likes to bind proteins, and so we need to basically block all the opportunities for the antibody to bind to the membrane and only allow it to bind where there's already a protein bound, and therefore only bind if our protein is there. So it's not just going to randomly stick to the membrane. So we block it by coating it with the free membrane with a generic protein. So often we use BSA or fat-free milk powder. So milk has this protein casein that's really abundant. And so it's nice and cheap and useful, and it'll block the background. If we're talking about a northern or a southern blot, then we typically coat with some generic DNA, so often salmon sperm DNA. With a northern or a southern blot, we typically do our blocking by coating the free membrane with a generic DNA. So this is often like salmon sperm DNA. Totally not joking here. We have a vial of salmon sperm in our cold room, or not the sperm, but the DNA from the sperm, nice and cheap and chock full of DNA. And then what we're going to do in any of these methods is we're going to wash. So we need to wash off any of the unbound blocking agents. So we've prepared our molecules, we have separated them, we've transferred them, we've blocked. Now it's time to send in our probes. And so remember, if we're doing a Western blot, our probe is going to be an antibody. And if we're doing a northern or a southern blot, we're going to have an oligonucleotide probe. In the case of a Western blot, because we typically have these two antibodies, this like two-tier system where we have a primary antibody and then we have a secondary antibody. So what we have to do is we have to do our primary binding antibody, and then we have to wash that. And then we have to do our secondary antibody, and then we have to wash that. So we have to do a lot of washing. And so it's really important, though, that we do a lot of washing, because we need to make sure that we remove any of the primary antibody that's not bound, because the secondary antibody doesn't care about what the primary antibody is bound to or not bound to. It's just going to bind to that primary antibody. That's all it cares about. That makes it useful, but it also makes it so that we risk having a false signal if we don't wash out all the primary antibody. And so we do a lot of washing, and we do our secondary antibody, and then we do a lot of washing, and then we visualize it, and we see how much of the specific protein is there. And your visualization strategy is going to depend on the probe that you used, and we'll get more into this in a minute. For the northern and the southern, at this point, you treat them basically the same, and you're going to probe them using a labeled DNA that complements the DNA that you're looking for, or the RNA that you're looking for. That's the only difference here. So your probe is going to be complementary to that sequence. You're going to send in this probe, and then you are going to wash off the free probe, and then you are going to visualize it. And so see how much of that specific DNA or that specific RNA was there and where it's located in the gel. So now let's talk a little bit more about some of the details with these various methods. So we'll start with the Western blot. So with the Western blot, we're dealing with antibodies. So in terms of our visualization, we're going to have to, these often have like an HRP. The secondary label is often this horseradish peroxidase. So there's actually this other little protein that is bound to this little protein, that's bound to this little protein, that's bound to your protein. And so this horseradish peroxidase, what it can do is it can actually convert an invisible chemical, this like 2-OPD, into this orangey brown chemical that you can see. And so after you bind the secondary antibody and you wash off the unbound stuff, you add this OPD. And if there's any of this HRB conjugated secondary antibody, this orange product is made. And that allows you to see that the other primary antibody was present. There's other label reaction options as well. And a lot of times the labels are fluorescent labels instead of one of those colorimetric label systems. And so those colorimetric, basically it makes a colored product that you can see with your eyes. With fluorescence, you're typically giving off some sort of light that you're measuring with the machine. And so the basic of fluorescence is that you have a molecule and it absorbs wavelength of one light, and then it emits wavelength at a different light of a different wavelength. And so you can shine one wavelength at it using a machine, and then it'll give off another wavelength that the machine then captures. And so obviously you need a more specialized machine in order to be able to do this type of thing, but you don't have to worry about adding in a bunch of other reagents. So it's simpler, but you have to have the machine to do it and that sort of thing. In the case of the DNA or the RNA, here you're going to have that radio labeled probe typically. And so it's really easy to make these probes. So you basically order a DNA sequence and from just like a company, because you don't have to have very long probes usually. These are going to be really cheap, just like the primers you would normally order. And what you're going to do is you can label their 5' end. Typically I do this with polynucleotide kinase. So you just add this kinase and you add radioactive ATP, and then it's going to, if the ATP was labeled, now your RNA or now your DNA is going to be labeled as well. And so here, you're going to have this labeled probe, and then you can use it as a probe. And how we actually visualize it is typically we stick, in the olden days, it was done more with like X-ray film type of stuff. Nowadays, we often use these phosphor storage screens. So basically these screens have this molecule in them that's going to be able to absorb the radioactive signal given off by the probe and kind of store it. And then what's going to happen is when we put it in one of these scanners, it's going to scan it and it's going to give it the little boost that it needs to actually give back that energy that it's stored as light that the machine can then detect. So if we place one of these screens on top of our membrane, then we can then see bands appear where our thing was located. When you are doing one of these, so often if you have like ethidium bromide stained your gel or something, you can visualize where the DNA or where the RNA pieces are. Although depending on how many there are, there could be like a big smear or stuff, but you could do that before you do all of this radioactive stuff when it was still in the gel and before you transferred it. Just a note, so you can then compare the location to where all of it was. Another note, so typically you're going to take your membrane and you'll wrap it in something like saran wrap, and then you put one of these screens on top. You put the screen on top with the white side down, like facing your thing. The reason why I'm saying this is because the first time I did it, I put it backwards, or maybe I think it was the first time I did it by myself. I put it backwards, and then the postdoc I was working with, basically called me stupid. And so, yeah, so don't feel stupid if you make mistakes, or especially if you haven't done things before. And so the white side goes down. Then depending on how faint the signal is, you trap it in this cassette, which is basically going to prevent the radiation from leaving the cassette, so it's safe. And then you can then later scan it. So those were the 3 main methods, but there are also other methods that are kind of riffs off of those techniques, because basically you can use all sorts of probes and all sorts of things. And so in the methods that we looked at, we were separating by size, but you can also separate by other things. Maybe you want to separate by charge using some sort of like isoelectric focusing. Maybe you want to separate by other things. So those are always options too. And those are going to be, though, you would still basically call it the same but you'd still be using the same sort of probes and the same sort of things that you were looking for. You can also go looking for different things with different things. So with an Eastern blot, you're looking for post-translational protein modifications. So things like phosphorylation, glycosylation, so sugar addition, and be lipidation, addition of lipids. And so typically these can be various probes, often antibodies against the specific modifications. So it's like a Western blot, except you're looking for modifications to proteins, not specific proteins themselves. In a Northwestern blot, you're going to be looking for proteins like you would in a Western, but here you're going to be looking using RNA. So you're looking for proteins that bind to a specific RNA that you put in. At the Southwestern blot, you're looking for DNA binding proteins. So it's like the Northwestern, except here you're using DNA to find DNA binding proteins. So I said for a specific one, but you can also look for like if it just binds any DNA. So others include the reverse northern. So here you're looking for DNA using RNA. So you'd have an RNA probe looking for DNA. So there's lots of different techniques is the basic point that I'm trying to get at. There's also like a Far Eastern blot, which looks for lipids. And so it actually uses a different separation technique, thin layer chromatography. So it's basically like this thin sheet that things move up. But the principle for all of these is that you're separating things by something, often by size, and then you are probing them with something that's specific for them. Although there's all these different techniques, there are still alternatives to some of these techniques. And so we still use the Western blot a lot, but we use the Northern and Southern less than were used in the past, because of like PCR based methods. So PCR is a way that we can make lots of copies of specific sequences of DNA using in like a test tube. We make these primers that bookends the regions we want copied. And then in PCR, we can have those regions copied. Often instead of a Northern blot these days, what people do is they do like qPCR, they do RT-qPCR, RT-PCR, basically reverse transcription PCR is the RT, or sometimes it's real time. But the basic idea is that if you want to make copies of DNA, you're going to want to, and you start with RNA, you're going to have to transfer it, you're going to have to copy it, make a DNA copy of it first. And so this is making DNA from RNA is called reverse transcription. So in reverse transcription, you make a DNA version of the complementary strand of the mRNA, and now you can make DNA copies of this. If you're looking for DNA, you start with DNA. So if you're looking for a Southern, like instead of a Southern, you could do this, you already start with DNA, and then the basic idea is that you could just make a lot of copies of it, and the number of, like the more you start with, the faster the signal that you're going to get is going to like rise. Often these are done with these like Tachman probes. So you have another probe that is actually going to bind in between the region you want to copy, and then inside the region you want to copy, and then when the polymerase goes to make another copy, it's going to break up this and then basically make it give off a signal. The more copies you make, the stronger the signal, and so the more you start with, the faster the signal is going to rise. And so this gives you a way of knowing the amounts of the thing that there was there. And so Southern blot is often now an alternative is to use a qPCR method, as well as for a Northern blot, you have to do the reverse transcription step first, but then you can do it. And so, but there are still reasons why you would want to do a Northern blot, or you would want to do a Southern blot even today. And so an example of when you would want to do a Southern blot is they're often used a lot. I know this is an old paper, but this technique is, well, I mean, older paper, not nearly as old as a lot of them I've talked about, but this technique is still used to study replication, so DNA replication. So when DNA gets copied so that a copy can get passed on to each of the daughter cells. When this happens, one of the ways that you can study this is with like this 2D electrophoresis. The DNA, you separate it in like two directions, and then you probe to see where a specific sequence is. These gels are really complicated, and I'm not going to try to explain them, but the shape of the curves and stuff that you see tells you about whether replication is occurring in that part. So whether there's like a replication bubble, like if you're in the middle of a bubble, if you're on the side of the bubble. So if you separate by size and by shape, then you can get these characteristic shapes when you're probing for various sequences. And so you can see, is the sequence in part of a shape that is basically telling you replication is happening? Is it telling you that no replication is happening there? And that sort of thing. And so you're probing with specific sequences. So you're doing like a Southern blot, and you're getting information that you couldn't get just by sequencing, because you're getting information about the actual replication state of the thing. So there are other examples too, where there's different types of separation that you can do, and then probe for specific things, and you're getting more information than you get just from the sequence. An example of when you would do a northern blot. These are used a lot in the small RNA research field. So small RNAs, like microRNAs, siRNAs, various small RNAs. These things are typically around 20 nucleotides long, and they're involved in RNA interference and all sorts of cool stuff, which I've talked way too much about in the past. Well, not too much, but I did a lot of work with, I've done my whole PhD with this protein AGO, which binds to microRNAs and brings them to targets to induce their repression. And I'm going through this really fast because it doesn't really matter for the purposes of this, other than to say that these RNAs are often, we use a northern blot to detect them, or I haven't actually done a northern blot, but I know a lot about them because they're done a lot in my field. And they're done because they can detect these RNAs well, and they can detect various variant versions of them. So when you are doing there's a couple problems with trying to do like an RT-PCR on small RNAs. One is that they're really, really small. And so basically they're the size of your primers. And so how are you going to copy them? So often this is done with like making specific adapters, and there's these various things so you can make copies of the region, but then you risk losing some of what was there. And also, if you are just looking for a specific sequence, you can miss variations in the sequence. So if you're doing like a qPCR, it's a yes, no, is this particular sequence here? But what if it doesn't tell you about what's next to the sequence and that sort of thing? So if there are modifications that are put on afterwards, like for these microRNAs, sometimes they get tailed, sometimes they get trimmed. And so you can see that if you probe for part of the microRNA, you see that there's these different lengths. So they have extra letters added on, or they have letters trimmed off. And so you can see this using a northern blot, where you wouldn't be able to see that with something like qPCR. So in addition to seeing the levels, you'd be able to see these different modifications on them. Another reason why you might want to use northern blot, in addition to small RNAs being hard, you can also, depending on what type of primers you're using for the reverse transcription step and that sort of thing, they can have bias as to what gets copied. And so you could have sequences that are present, but that are getting missed with the method. So some sequence might be getting preferentially copied, others might get lost in the process of your preparation. And so with the northern blot is that you don't have to worry about it being copied, you just have to worry about it being present. And then you don't have the bias that comes from the PCR technique and from the reverse transcription technique. So Southern blots have been used in some really groundbreaking, important genetic analysis, looking for specific DNA sequences and helping map the locations of mutations and the genes making different proteins. So a classic example is cystic fibrosis. It's caused by this mutation of this CFTR gene. And I have a whole post about this, so I'm not going to get into it too much. But they used Southern blots a lot because they were using probes to try to find a sequence that they found that was present in people with the disease, but not people without the disease. And they wanted to see where this gene, like where the gene for it was. And so they made all these probes, and then they were doing all sorts of probing to try to find the various, trying to find the actual gene, and then they were able to actually find it. They also did in northern blots to show that the gene was actually getting expressed in the cells of these patients that were most affected by the disease. So they would take this, the RNA that was made in the cells, like the messenger RNA, and then they could probe using DNA. And they were able to show that that gene was actually being expressed in those cells, which was important for showing that the mutation was in this gene. This gene was actually, this mutation and this gene was actually responsible for causing this disease. But even today, these techniques are still used.